so hopefully we remember this, is there's actually, here's the kind of real full balance one, 6 carbon dioxides, but plus 12 waters, plus light coming from the sun, right? Turns into glucose, C6H12O6, a carbohydrate, you should know, plus some oxygen, plus there's actually 6 waters that come out, but we use 12 over here, so really overall 12 waters went in, or 6 waters go in, because you get 6 out, and kind of see how that reduces down, okay? Really the idea is you have to put 6 carbon dioxides in to get 1 glucose molecule, okay? And that's where the carbons are coming from, big thing. So where does photosynthesis occur? Well, you should hopefully know that it occurs in leaves. And so if we were to take a part of a leaf and zoom in on this, if you remember back to all your little plant anatomy stuff, you have the epidermis layer up there. Okay, then you have this whole area, I'm going to scoot it over a little bit, of this whole thing is called the mesophyll. And you have the spongy and the palisades, the spongy with more surface area. And then you have your stomata, which we're going to talk more about in the next chapter when we talk about transpiration. This is where CO2 goes in, enters, and O2 and water exit. Okay. And water actually has to go up through the roots and all that kind of stuff. And then in these cells, in the mesophyll cells, each one of them is going to have tons and tons of chloroplasts. Okay, and that's a lot of them. And if you're asking yourself, what's this big thing? Hopefully you should know that's a central vacuole found in only plant cells. Okay, so they have lots and lots of chloroplasts all throughout their cells. Okay, and so now here's the question. If you're a prokaryote and you don't have a membrane-bound organelles, so you lack chloroplasts, no chloroplasts, how do you do photosynthesis? And the idea is actually they have chlorophyll, which is the pigment that absorbs the light and can turn that energy into other things, is built into the plasma membrane. So it's just built right in, no problem. They just use their plasma membrane for it, okay? But if you're a plant cell, you have a lovely chloroplast, which we're going to begin to draw here. I'm just going to keep drawing in green because, you know, well, plants are green. So here we go. This is my chloroplast, okay? And the chloroplasts actually have 2 membrane layers. They have an outer and an inner membrane layer. So here's the outer, and here's the inner membrane, okay? And this space inside the inner membrane is called the stroma, which is where the Calvin cycle takes place. That's part 2 of photosynthesis. Cycle. Okay. And inside the stroma, there are these little stacks, and they look like little pancake stacks, a lot of them. And they're basically, it helps increase surface area. Lots and lots. Each of these is a membrane, a phospholipid membrane. So you have lots of these stacks. And those, the big stack, the whole thing, if you were to do the whole thing, is called a granum. And what it is, it's a stack of thylakoids. Okay. And so these are the thylakoids, okay, and then inside the thylakoid space. And so we're on the thylakoid membrane is where the light reaction takes place. That's where light is absorbed. That's part 1 of photosynthesis. Part 2 is the Calvin cycle. Okay, so now we kind of know where we're going to go. We're going to zoom in on this light reaction in the thylakoid membrane first. Actually, we're going to take an overall view. Let's see what happens in photosynthesis. Look, and then we'll zoom in. Okay, so here we go. We're going to have light. I'm going to use red for light just because it's hard to see yellow on paper. So there's our light hitting. And so we have our what we call our light dependent reactions. And at this stage, water becomes oxygen, okay? So that happens. Also, we're going to make some ATP, and we're going to make some NADPH, okay? Which is what we call it. It's just an electron carrier. Carries around electrons. Think of it like a little train thing that carries around electrons. And then part 2 is going to be the Calvin cycle. And this is where carbon dioxide comes in. Okay, we haven't. And this is where we're going to get glucose is going to come out, C6H12O6, which you can't see me writing. Okay, and we're going to use these things up. We're going to use the ATP that we made, so we're going to turn it back into ADP and a phosphate, and we're going to use our electron carrier. So we're going to use that, and we're going to turn it back into NAD+. Okay. So it's kind of the gist of everything that we're going to do. We're going to see light being absorbed, making some oxygen, ATP and NADPH in our light reaction. And then in our Calvin cycle, we're going to use carbon dioxide to make some glucose using the ATP and the NADPH that's produced in the light reaction. Okay, this, just to reiterate, is in the thylakoids. And this is in the stroma, that space of the chloroplast. Okay. So let's now zoom in on the light-dependent reaction. This right here is actually the thylakoid membrane. That's what these like two lines are trying to represent, the phospholipid bilayer. Okay, so this is the thylakoid membrane. And what's the first thing that happens is we're going to have we're going to have this guy here. This is called photosystem 2. 2 comes first, and then before 1, it's because they were discovered in that order. Sorry, it's just how it is. And this one also is known as P680. That's the wavelength of light that it absorbs, the 680 wavelength of light. Okay, what's going to happen here is I'm going to have water come in. Okay, so here's H2O, and it's going to come in and it's going to actually drop off some electrons. And when it does that, it turns itself into oxygen, and it's going to also, okay, so here comes some, it's also going to pump some hydrogens, okay, across a membrane. So here we go, hydrogens going across. And my little electron, which we will follow in pink, my little electron is going here. Okay, and the reason why it does this, the reason why it has the energy to do this is because of my good friend, light from the sun. Okay, then what's going to happen is this little guy here is known as, I'm going to label him, so the electron gets passed over, he's known as PQ. Okay, it's just the name of him, don't worry about it. Then my electron is just going to be passed to what's called a cytochrome complex. Okay, so there goes my little electron, cruising over. Okay, and then it's going to be passed again. Here it goes, and it's losing energy a little bit as it does this to another guy called PC. So we go from PQ to cytochrome complex to PC. Then my electron gets passed over to this is now photosystem 1. Sorry, I always say that. So photosystem 1. The way I remember what order the photosystems go in is someone told me it's like 2, 1, and then like blast off, because we're going to make some ATP and we're going to make some other things. Okay, and this one's known as P700 because it absorbs light at that wavelength of 700. So here comes my light. Light provides the energy to re-excite my little electron. So here goes my little electron. He's all excited. Yay, electron. Okay. And so my little electron is excited, and I get to pass it to the next electron carrier. These are just little proteins that carry electrons. That's all they do, named FD. And then finally, I'm going to pass my electron, well, not finally. I'm going to give it to this guy here named NADP plus reductase. Okay. And NADP, you notice it's a reductase. It has that ASE, so it's an enzyme, and it's going to, you can probably figure it out, reduce NADP plus. Okay. And so that's what it's going to do. So this little guy here, this box is going to be NADPDPH, and it becomes that when it accepts an electron. Okay, before it was just, which has gotten passed this way. Okay, and so before it was just NADP plus, and when it accepts an electron, it also gains a hydrogen. It pulls a hydrogen out. Okay, there's something I haven't done with all these hydrogens, and actually I messed up. I drew the arrow the wrong way right here. I just noticed this arrow should actually go this way. We're going to pump a hydrogen. When those electrons move, we're going to pump a hydrogen. When the electrons move here, we're going to pump a hydrogen. And also I'm losing hydrogens over here. So what I've done is I've set up a little what's called a concentration gradient. You guys are familiar with this. I've added more hydrogens to the inside of my thylakoid than there are on the outside. So you're probably knowing what the question is going to be. Where do the hydrogens want to go? They want to go out of the cell. And you're going to want to draw a little guy that looks like this. Okay, kind of like a mushroom. And its name is ATP synthase. I bet you can guess what ATP synthase does. Synthesizes ATP. And the way it does that is it allows hydrogens to go through it, and when it does that, it actually spins like a turbine. It's kind of cool. It spins like a turbine, and we'll spend some time looking at this later, and creates ATP when it does that. Okay, so now, conclusion, let's sum up. Light's gone in, move some electrons along this little electron transport chain. That's what it's called. Okay, so just from one to the next, using the energy of light to excite my electrons. Pass my electrons to NADPH, and pass some electrons to ATP, and use the proton gradient to create some ATP. Okay, so let's see what's next for us. Now we're going to use that information to create the Calvin cycle, okay, or to look at the Calvin cycle, okay. So this is also known as a light-independent reaction, and this happens in the stroma. Now, this might look really complicated. The good news is there's very few things we actually have to learn in terms of crazy names, okay? So what's going to happen is we have 3 carbon dioxides coming in, CO2, okay? 3 of them we're going to have. And I know this is step 7, and we're going to pretend that because it's a cycle, so it's kind of hard to start. You just kind of pick a spot. We have these molecules called 3 RUBPs, okay? And what's going to happen is there's an enzyme, Rubisco, that fixes the CO2s. Basically what it does is it takes these RUBPs and these CO2s and combines them to form 6-PGA. Okay, I'm going to write a little list of the molecules you need to know down here. Okay, so down in my corner, molecules to know. Ready? RUBP, CO2, Rubisco, NADPH, ATP. Done. Okay, so I'm going to give you the names of the other ones. Don't stress yourself out. So then what's going to happen is we're going to use some of that energy that we made in the light reaction, okay, and we're going to put in that energy, and you can see that we've added these phosphates here, and you can color code them if you want. You can color them in little phosphates, yay. Okay, so here I've added some more. There's some phosphates added. Happy. Okay, great. And so now what's going to happen is my 6 NADPHs are going to donate that electron. Because remember I just said they're electron carriers. They want to get rid of it. They're over it. They don't carry that thing around. And they become 6 NAD pluses. Okay. And so now these things have rearranged and they turn into this molecules called there's 6 of them, DPGA, don't need to know that name, just whatever. Basically they're rearranging themselves, then they turn into this other one's called PGAL, great, who cares? And then what happens is one of those PGALs comes out at this point, and then actually what ends up happening is 2 of those PGALs will combine to form 1 glucose. Okay, because remember I only put 3 carbon dioxides in, and I said on the first page 6 carbon dioxides are required. Then I just have to regenerate my RUBP. I have to remake it. So I only have 5 PGALs over here, and then we're going to rearrange. We're going to turn them into some different kinds of carbons, and we're going to turn them into 3 RUPs, whatever. Basically we took them from a 3 carbon to a 5 carbon. Great news. Now I'm going to add some more ATP to the situation. So here's my more phosphate. So here's ATP coming from my light reaction, and turn it into ADP. And now I've remade my RUBP, which can combine with Rubisco Rubisco can combine my RUBP and with the carbon dioxide to make more PGAs. rearrange, add some energy, add some electrons, make some P-gal, take one of those P-gals out to make a glucose, keep the rest, regenerate our UBP.